Besides the nucleus, we do have some other membranous organelles inside of eukaryotic cells. The endoplasmic reticulum is one of the largest besides the nucleus. The endoplasmic reticulum is an extreme amount of membrane that's just folded back on itself so that you get a lot of what look like flattened sacs for this. Overall, it's a transport system. It does involve transport, but then it also has two parts, the smooth ER and then the rough ER. And ER is just a way to abbreviate the endoplasmic reticulum without having to say that mouthful there. The smooth ER, which is this portion here that doesn't have all the spots on the outside, that one is going to be responsible for lipid synthesis. So making of lipids, that would include fatty acids, triglycerides, maybe some steroids as well. And then the other type of ER that we have is the rough ER, and it's the one that you see here that has all of the little orange spots on the outside. The little spots are ribosomes. And so those ribosomes, they're going to be making proteins, and as they make proteins, they will be actually placing those proteins into the inside of the rough ER. And then the rough ER can take those and it can start to transport them. Now, working in conjunction with the ER is going to be the Golgi body. The Golgi body is pretty much right there next door to the ER, and it is going to be kind of the processing plant of the cell. It is going to receive whatever the ER sends to it, and then it's going to process that, and then it's going to package it up. And when it packages it up, it's going to produce these little bubbles, which we call vesicles. And so the vesicle is like this tiny little package. So it's a bubble, and it has molecules on the inside. So that's what's been packaged up, and that is now a transport container or cargo container that can be sent to other locations inside the cell. So the Golgi is taking stuff from the ER, switching it around a little bit, processing it, and then putting it into a cargo container that we call the vesicle. This is not something that's going to be found in all eukaryotic cells, but it is fairly common. Other sacs that we have inside of eukaryotic cells, there are lysosomes. Lysosomes function in digestion. We have peroxisomes, which function a lot in detoxification. So if there is a poison or something that's toxic to the cell, the peroxisome is going to help to get rid of that. And then vacuoles is another little bubble, or it can be a large bubble of membrane, and that one's going to have more of a storage function. It can be storage of food, it can be storage of waste, it might be storage of pigments, something like that, if that was something that you had a lot of inside of a particular cell type. Right here, we're just looking at kind of the coordinated activity of a number of these different membranous organelles. So here we can see the ER. Okay, so this is the ER, and we said the ER is responsible for making a lot of stuff, lipids, and then also working with ribosomes to make some proteins. We have the Golgi, which is shown here in the orange, and we can see kind of the pinching off or the forming of vesicles. Those vesicles would contain things that have been processed and are now ready to be shipped to some other cellular container. And then we also see the endocytosis, that engulfing, taking in of some different, in this case, a bacterial cell for the process of eating it. So this specifically is phagocytosis because we want to eat it. When it gets brought in, then we're going to fuse it with the lysosome. So the lysosome, just emphasize again, is for digestion. So if we need to eat something, the lysosome will fuse with that particular container that now has the bacteria, and then notice it immediately breaks it down into little pieces. And then if we want to get rid of those pieces, we can do what's called exocytosis, and exo means exit, so that would be one that is now exiting the cell. We do have some organelles that fall into a little bit of a different category, and what makes these different is that they're going to have multiple membranes around them. So they are similar to the nucleus in that respect because the nucleus had a double membrane, and these will have some extra membranes too. One of them is the mitochondria. The mitochondria, you can see it on the bottom here. The mitochondria has two membranes, and its membranes, the outer one is going to just be kind of a normal membrane. So if I draw it like this, and then the inner one, you can see it here, the inner one just gets folded a lot, kind of up and down. And what this does is it means that that particular membrane has a lot of membrane surface if you were able to just kind of stretch it out. So they've got these two sets of membranes. Overall, what the mitochondria is going to do is it's going to be producing most of the cell's ATP supply. And the ATP, that's the energy molecule or the currency that the cell is going to use to run its reactions. A unique thing about the mitochondria is that it does have some DNA and it does have some ribosomes inside of it, and that would be found in the innermost container right here. So inside this compartment, we'd find some ribosomes and some DNA. So the mitochondria can do some of its own protein synthesis because it's got some ribosomes kind of in-house, and it also has some genetic material inside of it. The chloroplast is another unique one. The chloroplast actually have 3 total membranes. If we were to draw their membranes, the chloroplast has kind of this oval shape. It's got 2 membranes that are kind of side by side like this, and then we have another membrane that's going to create these little discs inside of the chloroplast. The chloroplast is involved in photosynthesis. Not all cells are going to have chloroplast, but this is something that we do tend to find in plant cells and then many algae as well. There will be pigments specifically, which is why plants are green, and those pigments are going to be collecting light energy and using it to produce ATP, which again is going to be an energy source that can be used to run the cell. The chloroplast also has some of its own DNA and some of its own ribosomes, and that would be found in this area or that particular compartment inside of the cell. So if we talk about what makes these unique organelles, the mitochondria and the chloroplast specifically, this is thought to be because of the endosymbiotic theory, which is really this idea of how complex cells, eukaryotic cells, did evolve. So what this theory suggests is that very early on, so billions of years ago, eukaryotic cells evolved from a union of smaller prokaryotic cells. So you had multiple prokaryotic cells that unionized, kind of compiled together to make this larger structure. If we look here, we've got this initial cell right here, and notice that this cell, in this case, we've got an engulfing. So that would be an endocytosis that we're seeing right here. And then once we have that on the inside, notice how this is now going to have a couple layers around the outside. So you've got an extra membrane there that you didn't have before. That is thought to have been the beginnings of the mitochondria. And then it is believed that that happened again at some point, and that became the chloroplast. So we've got these smaller cells that were engulfed by endocytosis, brought into the cell, and then rather than getting digested and taken apart for food, they survived. And not only did they survive, but now that they're on the inside of the larger cell, they started to continue their activities in a way that benefited the larger host cell. And so endo means inside. Symbiosis is kind of a cooperation, mutually beneficial relationship in this case, because the smaller pieces that were engulfed were benefiting the larger cell, and then the larger cell was probably providing some type of a protection for the smaller. And then over time, it's believed that they became dependent on each other. There is a bit of evidence that supports this theory. One of the pieces of evidence, or a couple of them, we've talked about already. The fact that you've got multiple layers of membrane around these cells. And the multiple layers of membrane is what you would end up if you did have something that already had a membrane around it and you're engulfing it and bringing it in by endocytosis. Another piece of evidence is the fact that there is some DNA inside of these compartments, and then there's also going to be some ribosomes inside of them. So the DNA and the ribosomes are kind of remnants, it's believed, of what was present in that initial cell when it was engulfed and brought inside.